Thank you so much. Thank you for the accommodation and also for the kind introduction. My name is Wenjing, and thank you all for being here. Today, I would like to present a work recently from our group, from Dr. Xiangzhou's group, that we are trying to utilize the optimal transport analysis on spatial genomics data to perform the functional domain changes along time trajectory. We name our tool as spot. And we are currently still working on that. It's an unpublished work, so I will kindly ask you not to distribute it until the end of April, so that at that time I think I'm mostly finished with my work and wrap up my paper. So just want to give you some motivation examples, like why are we doing this? So the first example would be the cortical layer evolve along time trajectories. So in the brain regions, there are multiple layers of different cortical regions, like they have 6-layer structures with within each layer, they might consist of a similar cell type composition, saying a large proportion of neuronal cells and some non-neuronal cells like glial cells and oligodendrocytes, things like that. Although they have a very similar cell type composition, they might have different functions in each layers, and we want to identify that region or functional domain development along the time trajectory. And another quick example is also about the brain development, that in a higher level to see the brain structure, we might see in the original or earlier stage, there will be just 3 primary brain vesicles, and later on in the later development stage, it might divide into 5 secondary brain vesicles. So which means that there are certain functional domains evolve along time trajectories, which each functional domain would have a specific biological functions. So those are our motivating examples and what we want to actually detect along the time trajectory. However, as I mentioned, there are different layers and functional domains, and how to capture that. And thanks to the advanced technology, we might use this spatial transcriptomics techniques to obtain those information, like the domain information and the spatial gene expression matrix. So basically, this is a technique that profiles the gene expression on certain tissues of interest with the spatial localization information provided. Basically, it can be divided into 2 categories. One is this fish-based ones, and the other is the sequencing-based. They will all measure the gene expression counts, but differently, it's just the readout. So for the fish-based, it's just like the gene expression levels will be obtained based on the hybridization readout in the image, while for the sequencing-based data, they are just those reads mapped to the certain gene regions. And there are certain examples for that. However, even though there are certain difference of obtaining the gene expression, the data format would be the same, as one of them would be the gene expression count matrix and the other is a spatial coordinates alongside the each for each spot or for each cell. So here I would like to give a quick introduction of spatial transcriptomics data, which is measured by the 10x Visium data. So for this 10x Visium, each one would have a slide capturing the tissue of interest. And for that, there are several probes on that, where each has its coordinates, saying for this spot, it's just like 0 and 1. That spot would capture certain gene expressions located on certain tissues. And for this slide, it would capture approximately 5,000 spots. And for each spot, we will have those sequenced reads mapped to certain gene regions. In the end, as I mentioned, it will generate 2 different matrices. One is the gene expression matrix, and the other is the spatial locations. For this one, each row represents the spot, because for each spot, it might be very large so that it can capture multiple cells, or if the cell is large enough, it will just be the cell level. But for the 10x, most of the time we just call it a spot level, which means it would be an aggregation of a certain number of cells. So for this one, the row represent each cell or spot, and the column represent different genes. Within each cell and spot, we will have a spatial coordinates, which represent where the cell or spot locates. However, as I mentioned, what we were trying to do is that we try to identify the functional domains across the time trajectory. And thanks to this technology, the stereo-seq, it helps us to measure different spatial domains along the time trajectory. So from this example, they measured different 8 stages collected at different embryonic stages, and then they used the spatial transcriptomics data to profile each locations in each embryonic stage in each slice. So for this one, it will result in multiple count matrices. For example, we will have multiple gene expression matrix and multiple spatial locations accordingly. And each slice or each matrix is coming from each different time point. And in the end, what they try to do is that for different, like saying for the three initial domains, they will derive into organ specific, saying for brain, liver, or heart, things like that. So you will see a clearly certain of the split and certain domains might disappear due to the technology limitations, but it will reappear again in the later stage. But in the end, what we are trying to discuss is that whether we can use this kind of technology to detect those domains, whether about the disappearance, about the emergence, or the split of them. So here would be an illustration of that. So given all those spatial sequenced data collected from several different time points, when collecting all those profiles and the spatial locations, whether we can use a simple method to identify those domains and the development along the time trajectory. So please let me know if it's unclear or anything just pops up. Any questions are welcomed. So of course, there are certain existing methods in the trajectory analysis. For this kind of split, you might think of the single cell trajectory analysis, which are very popular in this domain, right? There are several tools like monocles, like PAGA, which utilize one or multiple single cell RNA sequencing data sets to identify the cell type divergence. For example, from the neuronal stem cells later on into the certain GABAergic neurons or the other inhibitory neurons like that. But for this one, the limitation is that they are only taking the single cell RNA sequencing data, but not considering the spatial locations that can be exclusively obtained from the spatial transcriptomics data. There are also other spatial trajectory analysis, for example, the ST-learn, the space flow like that. But what they did is that they tried to identify the pseudo time within one slice. So it only used one spatial transcriptomic data and only focused on whether they are certain pseudo time detected within this specific slice. So there are currently no methods designed for multiple spatial temporal transcriptomic data sets. Of course, there are certain methods specifically for the detection of regions utilizing multiple slices, but they are not using any temporal information. What they did is that they either try to integrate all different multiple slices or either remove batch effect or not removing batch effect. And then after the integration, they would perform certain clustering. or they will try to, based on the gene expression and the coordinates, they try to perform the image registration and alignment, and then they perform clustering. So either ways, what they trying to do is that they try to integrate all the slices together and then perform clustering to detect domains. In that way, it will not identify the domain difference, right? Because what they try to do is they try to detect similar structures, and also they will over smooth with the time trajectories because they try to drag all the similar structures together to make sure they are merged together. But that's not always the case, right? If one of the domains splits to others, then certain problems would happen. So in the end, we developed this kind of framework that we throw in multiple modules inside to achieve our goal. So basically, our framework would be an variational autoencoder based, which is a generative model that we incorporate two different modules where one is the SVGP encoder and the other is the GAT encoder. So this one captures more of a smoother pattern, and the GAT encoder captures more of a discrete pattern. So by combining them and aligning them, we will obtain a better domain results, and I will show you later. So we based on the latent space we obtain, we also add certain time constraints and clustering constraints to increase accuracy and to make sure that those domains are shared across the time points, but they are not kind of over mixed together. So let me kind of divide it into multiple modules to explain it a little bit more. I know there are many technical details, but I'll just try to explain it in a simple way. So for this one, when we use the SVGP encoder, what we did is that we try to build a kernel function based on the spatial locations. So basically, it will just be a distance matrix that calculated from all the spatial locations. And instead of the traditional VAE, where assumes that the Z would follow a standard Gaussian distribution, we assume that is conditionally on the spatial locations. And we use certain Gaussian process regression method to solve that problem to ensure that there will be spatial dependency inside this latent space. So the reason why I was saying this would capture a more or a smoother spatial patterns is because like we utilize all the spatial locations and we derive a distance kernel based on that. That means that we are trying to have this as a globally insight for the encoder to see. So here we do not do any thresholding or cutoff to remove certain dots because of the distance is too far away. Here we just preserve all those information inside this distance matrix. And you might say, like, if having this global patterns, that means there might be a lot of noises and it's hard to detect the local patterns that might split the boundaries. So that's where we accommodate that by applying another techniques into it so that it only captures some local patterns based on this k-nearest neighbor graph. So in this one, we try to construct the graph from the spatial locations, and we set the k to be very small so that each dot would only be connected to around 6 to 10 neighbors so that it will only see some local patterns and say these will be connected. And here we use another advanced technique in deep learning, which is the graph attention transformer. And different from the previous one, we assume the conditional distribution, but here what we did is that we moved the conditional distribution on generating the latent embedding. So where we utilize the gene expression information along with the gene graph information to derive the latent space, so that it still helps provide the spatial dependency, but not losing much information about that. I think I see a question in
Wenjing,
the
yeah, I'm just
chat.
going to read that to you. There is a question that just came in from Dana, and she wrote, for domain identification and development, is this limited to well organized slash defined these domains since samples must be coming from different individuals? And are there concerns that between individual differences or plasticity in these domains might be overcorrected even with the autoencoder approach?
Yeah, but I think I might need to ask like, what do you mean by well organized or defined domains from different individuals? Do you mean that those domains might not be shared across different individuals? Or, yeah, I'm trying to understand the question itself.
And Dana, if it would help at all for you to verbally have a conversation, I'm happy to. You can raise your Zoom hand if you want.
Yes,
But oh, hold on a second. Let me just give Dana the opportunity to.
sure, sure. Yeah, yeah.
Okay, Dana, I think you should be able to talk.
Okay. Sorry for the confusion. And this can also wait until later. It was more so thinking about fine structures or substructures that might be more difficult, have more variability between individuals, even in development. And just understanding the resolution that you're talking about here as far as domains. And then
I
related to that is more so the concern of if it's at this larger scale, are you, even with the autoencoder approach, that seems to have
I
improvements
see.
to this, is there a concern that you're missing out on possible real or interesting variability either between individuals or kind of over developmental time because of needing to be at that higher scale?
I see. Yeah, thank you so much for the question. If I understand it correctly, I'm thinking about you were asking about questions saying, for example, certain domains were detected here in the early stage, but it might be missed out due to the technical limitations, saying that maybe the spinal cord should be there all the time, but certain techniques might detect it and then miss it in the certain stage, if I understand it correctly. Yeah, so for that, I would explain how our framework works. But as you mentioned, yes, they need to be well defined because, like, for these ones, we are basically using the clustering technique to obtain those domains. So we need to know, like, beforehand, like, maybe approximately how many clusters are there or how many domains should be in the earlier ones. Then how many would be in the later ones. So that needs to be given. Those are the given numbers.
That helps
Yeah,
clarify.
but
Thank you.
that's a very good question.
Looks like we got a second question in. The question is, how could I apply that to the treatment of genetic retinal diseases in the field of genetic bases intraocular drugs? Any tips?
Yeah, I'm sorry, I'm not very familiar with this treatment of genetic retinal diseases. But I'm just thinking if you have those spatial transcriptomic data, as I mentioned, you might know like how many domains there should be inside certain time frame. And then I'm just guessing maybe you would have a baseline and then a treatment, like 3 days after treatment, 5 days after treatment, things like that. And then based on that information, you might use our tool to detect the domains. But as mentioned before, we might still need the number of domains that we need some prior knowledge on that. Yeah. I will definitely show that, for example, in the later real data analysis, we will show certain domain might disappear because of the limitation of the spots that can be measured by the 10x Visium. But yeah, I just want that might be giving you a clue. Yeah. Thank you so much for the question. Yeah, so the basic goal of developing this tool is actually for I would love it to work for the cancer treatment or something because those cancer regions can be clearly seen from the tissues. But yeah, thank you for those questions. That kind of reminds me of my initial goal. Yeah. So let's come back to the here that as I mentioned, I try to obtain different latent embeddings from these two different fractures, and then in the end, what I'm trying to do is that after obtaining the parameters from the variational autoencoder, I try to align them and connect them. The alignment is basically just for adjusting the skills learned from different infrastructures. And then after obtaining the latent space, what I did was try to perform a clustering method where we just simply use the k-means and we apply a k-means loss that we try to, for distinct domains, say for brain and heart, we try to maximize the distance between them so that we can better separate them. And then, of course, based on all the tissues collected from different stages, we are using this optimal transport loss, try to make sure that all the clusters across different domains which are similar to each other to be close to each other. So that's why we add those two different constraints to one is adjust within the time point, which is from the k-means, and the other is adjust across the time point, which is the OT loss. And another note for this is that we are kind of using a cluster level optimal transport loss. The reason is only because of the computational efficiency, because if we are calculating the cell-wise optimal transport matrix, it will take a long time. But we notice that when shifting it to cluster level optimal transport loss, it does not sacrifice that much accuracy, but it can largely accelerate the efficiency of our method. So that's the main reason for using it in a cluster-wise way. So here I would like to show you some simulation study where we obtain this kind of structure from the Ellen mouse brain from the embryonic stage 13.5, and they give the annotations for different brain regions. So that's why I directly use that. I perform some image analysis to extract those boundaries, those domains, and then I try to simulate this procedure of three brain vesicles emerging into five along the time trajectory. So basically the simulation settings would just be, for this one, we consider them as the forebrain, midbrain, and hindbrain in the first time point, and then later on the forebrain splits into two telecephalon and the diencephalon, and then later on the hindbrain split into two cephalons later on. And for each region, we are just simulating four different cell types where we have excitatory neurons, inhibitory neurons, astrocytes, and OPCs, and accordingly we use these cell type proportions. And for specific simulation details, when we have three domains in the first time point, each domain would consist of these four cell types with the specific cell type proportions. And when a split occurs, we would assume there will be, say, X percentage of D genes happening between these. For example, between this red dot and the purple dot, there will be 20% of neuronal D genes there. But if there is no split, I'll just assume there is no D genes there. But for along the regions, we will assume there will be Y% of neuronal D genes. And the reason why I specifically said it to be neuronal is that I read this paper published by Dr. Yao in Nature, that they mentioned that neuronal cells can be more regional specific compared to the non-neuronal cells. So basically, I'm just trying to mimic the real world. I know it's not a perfect scenario because we are not mimicking the D genes along the trajectory, but here I just want to give you a quick flavor of how our method works based on this simple simulation settings. So we simulate 4 scenarios from the easiest to the hardest, where they have more D genes in the first than the last 2 scenarios, because if there are less D genes, it's harder to detect. And here is a quick example of the simulated data. So if you are looking from a cell type version, which means that because each domain consists of these 4 cell types, although they have certain number of the genes along the development, but if you try to see from a cell type annotation, you will see it's really just a mix of all the cell type information. But if telling from a domain, you would see that each one would be separated into domains. So here just a ground truth to show that from a cell type wheel or from a functional domain wheel, how would these brain vascules look alike. And here I would like to present our simulation performance about the clustering accuracy, which means the domain detection accuracy. And we use the ARI and the homogeneity here as an example. And the ARI is kind of measuring the similarity between the 2 clusterings of the ground truth and the predicted domains. And the homogeneity is for saying whether each detected domain contains just one single true class. So here, the reason why I use average is because we use multiple replicates. So for each scenario, we replicate it for 10 times. And also each scenario consists of three results from different time points, right? So that's why we calculate the average of it and then plot the box plot of the 10 replicates. So this is the average of 3 time points from 10 replicates. So as you can tell, a higher ARI and higher homogeneity indicates a better performance. And the first one, the yellow one, box come from a spot dot, and all others are the 10 different methods developed for the multiple slices spatial domain detection. And we can see a superior performance from our spot dot. And as I mentioned earlier, the first 2 scenarios are the easier scenario, and the last ones are harder. So we do see a little bit performance drop for the scenario 3 and scenario 4, where less D genes are there, but we can see like other methods maybe does not perform that well, or they just drop a lot more based on different number of neuron D genes across the split or cross region. And we also try to use the PAS and the chaos, those 2 different metrics to characterize smoothness, because in our simulation it's pretty simple and the domain boundaries are pretty smooth. So we can use this kind of PAS and chaos to identify whether they are like random spots located outside of the certain spatial domains, or whether they are preserving the spatial continuity. And for these 2 metrics, the lower the better. So you can see that it's still for spot dot, which is the first yellow box plot, we do much better than other methods. And here I want to show you a quick example of this most difficult scenario, that if the ground truth is like this, and how we perform in this most difficult scenario. So I just use one replicate as an example, saying that the ground truths are these domains, and we can see spot dot perfectly define those domains split along the time trajectory. But if checking the other methods, these 2 are also like top performers, which is the second and the third performer. We can see that spot VAE is hard to detect the split in the third time point. And for the spot align, it can only detect certain big regions, but considering they are some certain just random dots in there as different domains. So that's absolutely not true. And I'm also using another method as a kind of example of underperforming example that for this one, it basically just detects the cell type information instead of the domain information, as I showed earlier, that if looking from a cell type perspective, it will be just randomness in this simulation data. And from the embeddings, we would also see that if here is the ground truth, I'm just trying to use this to illustrate that this one is the first, like the forebrain, I would say the forebrain here in the ground truth, and then later on it developed into two different domains which are closer by, which means this one is the telecephalon and this one is the diencephalon, things like that. But if you look at the spatial line, it will more identify the cell type information and rather than this kind of clear pattern of the ground truth, which means that they try to merge all the time points together to make sure they illuminate the batch effect because they consider this as batch effect, so they try to remove it as much as possible. That means they result into a mixture of time point. But in our case, we are not mixing them together because we think the time point is still a kind of a difference, and we debate to remove that in our method. So here is a quick example of the domain split. So here are two scenarios. One is the easiest and the other is the hardest. And we can see that maybe it's a little bit blurred, but what I'm trying to convey here is that from the optimal transport analysis based on the embeddings we obtain from our framework, we can absolutely see that this T00 is split into two with a very confident transition probability, and also same thing for the hindbrain region, and similar for both two. So I think I see a question here based on the Euclidean distance BW with.
I'll read it out loud
clusters.
if you want. So the question is, your OT loss is based on the Euclidean distance between clusters in the latent space, correct? Does that mean you're mapping all clusters from one time point to all clusters in the previous time point, and you repeat this across all time points? Are there issues with unbalancing, such as when there are more clusters in one time point than the other?
Yeah, thank you for the question. Yes, my OT loss is based on the Euclidean distance between clusters. And here we just use the cluster centrist to perform the OT loss is because that we are trying to speed up the whole procedure of calculating and the loss constraints. So here we are just using saying if we have those like cluster centroid, we are just using the cluster centroid to perform the OT loss. Does that mean you are mapping all clusters from? Yes. So I'm, as I mentioned, a number of clusters are pre-given to the tool if we do the analysis. But if we are not doing the analysis and we do not know the number of clusters, what I did was that from the embeddings, I'll just perform k-means clusters with k equals to 10 or 20 so that we can also capture all the clusters from the domain. And for that OT loss, we just compute 10 to 10. So that will be a fixed number of clusters. So from the embeddings, I'll obtain 10 clusters from time point 1 and 10 clusters from time point 2, and then I'll try to perform the OT loss between the 10 clusters and the 10 clusters. Are there issues with unbalancing? Yes, that's a good question. And for that one, during the training, I don't see a problem because I set this K to be relatively large so that it can still capture numbers of different clusters. Because, you know, if your clusters are not well separated, saying I have 10 clusters for this time point, right? But there are only 4 of them, I would assume that certain maybe 2 of the clusters would reside in the same cluster and 2 clusters would reside into this cluster and 3 here, 3 here, things like that. So that would not affect that much because we set that relatively large. But after the training procedure, when we finish all the training and we obtain the final embeddings, we would have that questions here, and we need the user to pre-input the number of clusters they want for each embeddings, so that we will see we will perform a cell level optimal transport analysis and then aggregate into the clusters based on what user input is. Is that clear? Yeah, I know it's a little bit confusing maybe because during the training, I'm using the OT loss from a cluster level, but from the downstream analysis after the training, I'm also using the optimal transport to obtain this transition matrix, but it's from the cell level and we can define different number of clusters. Yeah, thank you. So here are some results from the simulation settings, and I would like to provide more on the real data analysis. So the first one would be the one that I mentioned, that due to the technology limitations, that certain the 10x Visium can only capture 5,000 barcodes, and also the slides would be limited to certain size. So that's where for this mouse embryonic development, we would see at earlier stage, it would capture more of this mouse embryo, saying that the brain region, the heart and liver, and later on it will be capture only part of the liver. And then in the 15.5 embryonic stage, it will be only focusing on the head and ignoring the heart or liver. So that's where things happen, that certain regions would appear and then it disappeared due to technology limitations or certain other limitations. So what I here, I just also want to show you a quick performance chart saying that our method outperforms, and here which each dot represents for the average from the 3 different time points, and each one would be labeled by this different shape. And we can see that spot dot also, the average of them also outperform other methods. And I would like to give you a quick idea showing that how spot dot results compare to others. So for example, here we can see the spot dot can identify those boundaries between the heart and liver and also in the second time point. But for the 2 competing methods, which are also having a high performance, but they either can only find these boundaries here, or they might lose certain of the information and they cannot detect the boundaries in the second time point. I do notice that we obtain certain region split here, and that is basically caused by the numbers decrease in this, because in the original one they also think they detect certain liver, but I don't think that's the truth, because as you can tell, for liver is the green dots and it's kind of spreads all over the place, and I don't believe that's the true liver organs. So what I'm trying to say, another thing I'm trying to discuss is that this PACE 2 method is the one I mentioned, using the alignment and do the clustering, but you will find out that when trying to do the image registration and also the coordinates alignment, they cannot actually alignment because they need to either shrink this the image from the 3rd time point, or either try to enlarge the first 2 time point, which they didn't do for their method, and they result in a pretty messy alignment and does not align those well. Yeah. So another thing I want to mention is that we do observe the liver disappearance after having the optimal transport transition matrix. I'm trying to focus on only those ones are larger than probability with over 0.2, and I plot this out. So we can see that for this liver region, which are the green ones, they disappear and cannot be detected in the 15.5. But as a compromise, as I mentioned, because the number of clusters are given, if we are given 4 here, then we might not have many problems, but we are still given 5 here based on the original ground truth, so that we can have a better comparison on the performance. So here what we get is that we have certain regions that kind of aggregate with the heart region, and also we denote split here in the mesenchymal region. But later on, we justify that these are all detected, like these regions for this tongue and muscle regions, they are all detected by all the methods. And also from the stained image, you can clearly see this here. For that, and using the gene enrichment analysis, we found that for this cluster 3, they are a combination of like a muscle system process, but later on they are more specifically saying the cluster 0, which is the darker color here, they are more focusing on the muscle development, and that one is more focusing on the bone development. And another thing I want to mention is about besides the domain disappearance and the domain split, we also maintain a very good domain consistency. Saying here for this blue domains we detected, we perform the gene enrichment analysis, and we do notice that there are certain common biological processes presented in all 3 time points, which are the forebrain development and correspond well with the stained image as well as the location they are there. And we also perform a Venn diagram for overlapping the gene enrichment for those 3 different clusters. And we do notice there is a high overlap between 3 of them, where we perform a permutation test. The p-value is significant. And we also noticed another interesting thing is that for the earlier stage, there are not many functional time point specific gene enrichment terms, but later on for the 15.5, they are more of them indicating the regions are more specified from my understanding. Yeah, I think I see a hand raised. Oh, is it by mistake?
I don't see one. I think one flashed up, but I don't actually see one.
Oh,
Yep,
okay.
the
Misclick. Okay, got it. Okay. Yeah. So I will I think it's maybe around the time, and I do have another questions I want to maybe say more about is that, as I mentioned, the SVGP captures more of a smoother pattern, and the GAD captures more of a discrete pattern. So by combining these 2, we obtain the superior performance we have. And we also perform some additional analysis, but I think due to the time constraints, I will not show that. But basically, the idea is that from this stereo 6 sample, it captures different stages of axolotl brain development. And from their paper, I noticed that in the earlier stage, the MP and DPA is hard to discriminate, but later on, it kind of evolves to a more easier detected ways. And we also want to show that we perform good on the domain consistency for this ventricular region. And also we want to say that we have detect this pallium split. But as I mentioned, it's not very perfect, that it cannot detect the boundary between the medial pallium and the dorsal pallium, but it does show certain indications that there should be a split there. And I'm just trying to say that our method aggregates those related regions across time point together compared to those regions given by the original ground truth, because they are just aggregating those ones from the same time stage together. For example, here they are from the last stage and they group the dorsal and lateral together. But in our analysis, we are trying to we aggregate the lateral pallium from across time points. So maybe in the end, I'll just go with a quick conclusion that we developed this spatial to identify these functional domains for data collected from multiple time points, which is for the spatial temporal time point, and we want to provide insights in the domain development along time trajectory, either the disappearance or emergence or split. And we try to synergize the global and smooth pattern and also local descriptive patterns either from the SVGP and from the GAT to achieve a better domain detection. And we use this probabilistic deep learning model, which is the VAE, so that we have the hope that it can be further used for generative purpose, so that maybe we can perform some simulation data to use that to as a real data simulator. And the last thing is that we add those clustering constraints and time constraints to increase our domain detection accuracy. So I think it's around the time, and I want to leave a little bit 5 more minutes for questions. And here are the acknowledgements that all the people from Dr. Joe's lab, and also we have 2 collaborators on this paper. And sorry, I think Marcy, I think you're muted. Yeah, no, I think you might read. Yeah.
Thank you. I'm so sorry. I thanked you and everything, and nobody heard it. So yeah, thank you so much for this wonderful talk. We do have another question come in. And the question is, first I just say thank you for the talk. And then how is the distance kernel and genomic profiles given to the VAE? Is it vectorized, or is the structure of the distance matrix preserved by the neural network architecture? Also, what do you think of extending this to learn continuous trajectories? If I understand correctly, right, now it seems like a discrete sequence of OT maps are inferred.
Yeah, thank you so much for the question. Yes, your understanding is correct. So I will try to explain it one by one. So for the distance kernel and the graph, we are actually using 2 different frameworks. One is for the distance kernel, I'm giving it to the SVGP encoder. And for the genome profiles, we just vectorize them into each sample or each spot, I would say. Maybe spot is a more accurate word for that. So for that, we will give this vectorized genome profiles from one spot, and then we add it to it. But here, the distance kernel would be added as a constraint on all the latent space. So as I mentioned here, this would be after learning the latent embeddings, we would enforce this kind of spatial locations, the distance kernel here as a conditional distribution. So there involves a lot of techniques in this SVGP. So if you are interested, I would highly recommend you go over this paper here. It's the JSBEC from published in AI Stats. That's how they try to use the Gaussian process to model the spatial locations inside this, along with the VAE encoder. And the other one for the graph, we are using a separate encoder, which is the graph attention transformer, as I mentioned here. So these 2 are completely standalone, but the input would also be a vectorized genome profiles along with the graph built based on the spatial locations, right? So for each node, it will have a k nearest neighbor, and the graph will be fed into the graph attention transformer. And for each 2, they will derive the mu and the sigmas for the VAE, and then I perform the alignment. And do you think of extending to learn continuous trajectory? Yes. As I mentioned, our OT actually is, discrete to just perform some loss constraints. But I do think it can be used into continuous trajectories. Saying I know there are some ongoing work that try to just using the graph neural network to model the continuous of the time point. Right now, what we did is that we just we just have those time points learned by these 2 different encoders. But I do think with the current technology, it's possible to do that. And another thing to mention is that for this OT, I'm using the Waddington OT, which can also measure the cell growth, where we were thinking about to add it into our next following paper, saying that we might model that later on. Yeah, I hope I answer your questions.
At the moment, there are no other questions, but we can certainly give it another few seconds to a moment just to make sure no one else is typing at the moment. We'll just give it a
Yeah,
moment.
sure.
All right, well, I'm not seeing any other questions having come in. So thank you so much, Wenjing, for this wonderful presentation. Really, really interesting stuff. And thank you everyone for attending today and for the excellent questions that you asked. And hope to see
Yeah,
some of
thank
you.
you so much. Yeah,
Hey,
thank you so much. Yeah, thank you for your time and invitation. Bye.
bye, everyone.